All right, now I'd like to talk about these neurotransmitter molecules, right? So remember, we have neurotransmitter being released into the synaptic cleft. What is the nature of these chemicals that are being released into the synaptic cleft? What are neurotransmitters like? Well, it turns out that we have a huge diversity of neurotransmitters, and they fall into a variety of chemical classes. I'm not going to expect you to know any of this, but I just kind of want to give you a sense, right? So these are some examples of neurotransmitters: acetylcholine, which is a catecholamine, as you learned before. We have some amines. We've got just simple single amino acids. So glutamate is actually kind of the primary excitatory neurotransmitter in our brains. GABA is like a modified amino acid, and it's the primary inhibitory neurotransmitter in our brains. And then we've got a couple other: adenosine, nitric oxide, which some people don't really consider to be a neurotransmitter. It's kind of more of a like neuromodulator. But in any case, the main point here is that we have a huge diversity in terms of the types of molecules that we have that are carrying these signals from one neuron to another neuron. Another thing that I would like to draw your attention to is that we have a variety of molecules, both naturally occurring or chemically synthesized, that either mimic or block the action of these naturally occurring neurotransmitters. So I just kind of want to give you a sense for what we, some of the things, some of the kinds of molecules that we have acting as agonists or antagonists for our neurotransmitter receptors. So nicotine is an agonist for acetylcholine receptors. Actually, the type of acetylcholine receptor was named after nicotine, so we call them nicotinic acetylcholine receptors. Curare is like it's used on poison darts, right? So that actually acts as an antagonist for acetylcholine receptors. Alpha bungrotoxin is black widow spider venom. So that blocks acetylcholine receptors. And I just want to say that we have acetylcholine receptors on our skeletal muscles. So these guys are going to affect our ability to contract our muscles. Just a few others, you might notice LSD. So some of our like recreational drugs are going to act on these neurotransmitter receptors. We've got antihistamines here, alcohol and barbiturates. So potentiators are not exactly agonists or antagonists. They are, they kind of enhance the effects of the naturally occurring neurotransmitter. Okay, so that's just to give you an idea. I don't expect you to memorize any part of this table at all. It's just to give you a sense of what's out there. Okay, the other thing that I want to point out is that one neurotransmitter can bind multiple receptor types, right? So you see glutamate here, and it's got like these three different kinds of receptors that it binds. And I want you to take a moment now and think about what the implications are of that. What does it mean? What is it going to mean for the body when we have one neurotransmitter that can bind multiple receptor types? Okay, so hopefully you were able to figure this out. We've gone over this before. If you have a neurotransmitter binding multiple receptor types, those different receptor types are going to trigger different signal transduction cascades and lead to different effects in their target cells. Okay? All right. Let's move on. Okay. So last thing that I want to say in this section is I want to address this question or this issue of how we encode different strengths of signals in the nervous system. Okay, so you might remember, hopefully you remember, that action potentials are all or none. You always get the same size action potential no matter what, well, you either get a full-size action potential or you don't get any action potential at all, which means that we are sort of faced with this problem of, well, if the action potentials are always the same size, how do you encode different strengths of signals, right? You can't do it in the size of the action potential. So it turns out that the way we do that is we do it through action potential frequency, which then translates to the amount of neurotransmitter that gets released from the neuron. Okay, so let's take a look at this figure, and I will explain this concept using the pictures here. Okay, so I'm going to draw your attention to the bottom first. Okay, what we're doing, what you're seeing in these plots up here is what is happening in 3 different locations on this same neuron. Okay, so we're recording from 3 different locations on the same neuron, and then we're also going to look at the neurotransmitter release that we get from that neuron. Okay, so looking at the top here, you can see that we're starting with a sort of moderate-sized graded potential, close to right here. Okay, that's going to translate into a few action potentials, which then travel down the axon, and that results in some neurotransmitter release. Okay, looking at the second panel here, we are starting with a larger graded potential, and you can see that that's going to translate into more action potentials, right? So we have a larger number and a higher frequency of action potentials. Those travel down this cell, and that leads to a greater amount of neurotransmitter that is released, okay? And then that then is going to indicate to the postsynaptic neuron, hey, we're getting a bigger signal here. And the postsynaptic neuron is going to respond to that bigger signal, all right? Okay, so that wraps up this first little video about presynaptic vesicle release. Here are your learning goals. Here's your vocabulary. See you next time.